So now I would like to introduce our webinar speaker, Dr. Laura Weber. Laura received her PhD in bioengineering from the Karlsruhe Institute of Technology in 2017 under the supervision of Professor Frank Breitling. During her PhD, she dealt with the development of assays for the investigation of antibody fingerprints with high-density peptide arrays. In her studies at the KIT, she specialized in microsystems for life sciences, medicine, and biomolecular separation engineering. Currently, Laura holds a postdoc position at KIT and focuses on different projects for the application and fabrication of peptide arrays. The title of the presentation is Deciphering Antibody Fingerprints Against Pathogens with High-Density Peptide Arrays. So once again, please use the questions pane to type your questions. And again, we will share the slides and post webinar file with you and also answer all your questions. Laura, so I'm handing the cursor over to you. Please take it away.
Yeah, thanks for the introduction, Carsten, and thanks everyone for joining. So in my talk, I will first introduce you to the two articles that I will talk about today. After that, I will shortly talk about the peptide array technology. And after that, about the two techniques that we use for our studies, namely epitope mapping and substitution analysis. After that, I will present the results of the two studies in which we compared the antibody fingerprints of different individuals. I will end my talk with a conclusion and a short outlook. So the first study that I will talk about today was published in 2017 in Biotechnology Journal, and the title is Identification of a Tetanus Toxin-Specific Epitope in Single Amino Acid Resolution. This study served as a model system to establish the method due to the high vaccination rate against tetanus. Subsequently, in the second study, we investigated antibody fingerprints of Lyme disease patients against an immunodominant antigen of B.burgdorferi. This study was published in Engineering and Life Sciences also in 2017. In both studies, a well-established method for the investigation of antibodies was combined with a new step to increase the resolution of the specificity. To investigate antibody specificities, we must look at the antigen-antibody complex. Antibodies bind to the antigen simplified according to the key lock principle. If the structure fits very closely together, molecular interactions come into effect. Therefore, antibodies can be identified via the binding site, which they recognize on an antigen. The antibodies hereby only interact with a small part of the complex antigens. In this case, it's colored red, and these so-called epitopes can be imitated with peptides in the lab. If these peptides are arranged in array format, you end up with a peptide array, which you're probably all familiar with. For both studies, we used peptide arrays that were synthesized by pepperprint with a spot density of 800 peptide spots per square centimeter. In order to investigate the antibody response against known antigens, an epitope mapping is carried out. Therefore, the linear amino acid sequence of the antigen is cut in silico into overlapping fragments. Subsequently, a peptide array consisting of these fragments is synthesized and incubated with the serum of a patient and a fluorescently labeled secondary antibody. A fluorescent scan afterwards shows which peptides an antibody has interacted with. All experiments were carried out with arrays that contained 15 mer peptides in duplicates. In both studies, the entire antigens were covered with a lateral shift of one amino acid. So the process of epitope mapping allows for the identification of peptides or epitopes with an antibodies are interacting with. Theoretically, however, different antibodies could bind to this peptide and vice versa. An antibody could also bind to different peptides. It is therefore necessary to resolve the specificity more precisely, which is possible by substitution analysis. All amino acids of one peptide are successively replaced by all others. The rest of the sequence remains preserved. Schematically, such a substitution is shown here with a 3-mer peptide and 4 possible amino acids. So in each row, another amino acid is substituted. The scan of a complete substitution analysis can be seen here. This is an example of the tetanus study. Vertically, the original amino acid sequences show. Horizontally, we see the amino acids that were substituted in the respective position. The first rows show that the signal, so the binding of the antibody to the peptide, gets not lost by the substitution, which means the positions are not binding relevant. Only in the case of glutamic acid, the signal is lost through the substitution by all other amino acids. So this position and amino acid is binding relevant. In the case of the following amino acids, the position is relevant. However, the respective amino acid can be replaced by chemically similar amino acids without losing the binding capability. The substitution analysis increases the resolution of the specificity, and furthermore, it enables bioinformatic analysis. So identified motifs of binding relevant amino acids can be cross-checked against databases to identify antigens from other pathogens that share the motif. By these bioinformatic analysis, cross-reacting antigens can be identified, and therefore false positive test results of diagnostic tests can be excluded or investigated. So I come now to the results of the 2 studies. In the first case, we used the vaccination against tetanus as a model system. The antigen is in this case the extracellular tetanus toxin. The toxin, which can be seen here in its 3D structure, is produced by bacteria called Clostridium tetani. It consists of 2 chains. The heavy chain is responsible for the uptake and transport into nerve cells. The light chain has the actually toxic function of preventing the release of neurotransmitters and thereby triggering the typical lockjaw. In order to investigate the antibody response with peptide arrays, an epitope mapping was carried out with the sera of 19 vaccinated individuals. Vaccination titers were confirmed by ELISA. The original fluorescence scan of an array is shown here. Enlarged is an area that showed a signal for multiple sera. Around each array, a control frame of a polio epitope was synthesized. The enlarged area shows a band of successive peptides as it's typical for an epitope. The range of key amino acids with which the antibody interacts is shorter than the peptide itself and therefore passes through the overlapping peptides. The binding relevant area of this peptide is marked yellow. As soon as all important amino acids are present in the sequence, an interaction takes place and the peptide shows a fluorescent signal. So this figure here shows the result of the epitope mapping for all sera that were examined. In principle, we see the same as on the previous slide, only in this case, the peptides are no longer arranged in array format, but in a line one after the other from left to right. Also, the fluorescence intensity of the peptides was evaluated and is displayed in a color gradient. Each line shows the result of a different serum. In total, 1,315 peptides covered the sequence of the toxin. As we were interested in similar or coincident antibody responses, we here show only the part of the heavy chain on which we discovered such similarities. In all other parts of the heavy chain and on the light chain, the sera showed very different results, and these differences might indicate that conformational epitopes play a role. Due to the consistent antibody response of 8 patients in the marked region of the heavy chain, the first peptide of the band was investigated in a substitution analysis. So the substitution analysis was used, to our knowledge, for the first time to compare the antibody specificities of different individuals in this accuracy. On this slide, we see the scan image of the substitution analysis of one serum and the evaluation in form of a heat map. So basically, the color represents the staining intensity of the peptide. But the evaluation of the fluorescence intensities in the substitution analysis was done in relativity to the original amino acid. This means that in each row, the intensity of the peptide with the original amino acid is set to 100%, and the intensity of the remaining peptides is set in relation to this. From a relative intensity of 60%, the field is colored in red. The result is a heat map which reveals the characteristic antibody fingerprint. The heat maps of 2 further sera are shown here as examples. The results of all 8 sera that were examined are compared in tabular form on the next slide. So in this case, the original sequence of the substituted peptide is shown horizontally. Below this, we see the binding relevant amino acids and those that can be alternatively synthesized without losing the binding. X means that this amino acid end position are not relevant for the binding of the respective antibody. Each row now represents the results of one serum, and it is clear that the specificities of the different sera are almost identical, and that alternative amino acids often agree, especially if they are chemically similar, as we see it for the hydrophobic valine and isoleucine. This result means that the antibody specificities, despite their random development in the body and the immune system, reach a very similar state if the same epitope is detected. In order to confirm the interaction of the identified antibody species with the folded toxin, we isolated the antibody from the serum of an individual by affinity chromatography. The first peptide of the epitope served as the ligand. The 2 fluorescent scans show the control incubation with serum prior to the isolation and with the isolated antibody. So we clearly see here that the isolation was successful. The ELISA to detect the interaction of the isolated antibody with the folded toxin was positive. Furthermore, we validated the antibody-peptide interaction on a second platform. We used maleimide surfaces on which we spotted 4 different pre-synthesized peptides. The first peptide was a positive control, which we identified in a different study. The second peptide was our identified and substituted epitope. The third and fourth peptides were negative controls. And in each of these 2 peptides, we exchanged 1 amino acid that was identified of being relevant for the binding. So in the first negative control, the valine was substituted by alanine. In the second, the lysine was substituted by arginine. The left image shows the incubation of such an array with a serum that was negative for our identified epitope. This serum only shows interaction with the positive control peptide, and on the right, we see the result of the positive serum interacting with the positive control and the identified epitope. In both cases, the negative control showed no signal. To validate the knowledge of the identical fingerprints, we have investigated the antibody response to a more complex pathogen with multiple antigens. Borrelia burgdorferi, a spirochete transmitted by ticks and causing Lyme disease, is such a pathogen with several antigens. One of these antigens is the VlsE protein, a variable surface protein. The VlsE antigen is highly immunodominant. This means it triggers an antibody response in many patients and is suspected to play a role in the chronic form of Lyme disease. As in the study before, we first performed an epitope mapping to obtain an overview of the antibody response. In this case, 358 peptides represented the whole antigen. The results are presented in a similar way as in the previous study. In total, 17 Lyme disease positive and 7 negative sera were screened. In this study, different epitopes were detected, but a trend could be observed that either epitopes at the N and or C terminal end of the protein or at the variable domains are detected. The 5 areas highlighted in color were examined in more detail in substitution analysis. So in this case, the substitution analysis showed a similar picture to the previous study. Shown here are an original scan of the epitope mapping, the scan of the substitution analysis of the highlighted area, and the evaluation of the fluorescent intensities as heat map. The substitution analysis showed that the binding relevant amino acids are almost identical. However, in this study, we observed more differences in the alternative amino acids. To clarify the position of the fingerprints on the antigen, we have colored the corresponding amino acids on the 3D structure. Orange and green are the epitopes and the N and C terminal. The other 3 epitopes are located on the variable domain. The variable domain of the antigen is interspersed by constant or invariable regions, and all binding relevant amino acids turned out to be part of these interspersed invariable regions. However, we did not investigate all variable polymorphisms of the antigen. For each of the epitopes, the relevant amino acids are preferably located on 1 or 2 sites of alpha helices. The bioinformatic analysis of these fingerprints has shown that the 3 longer fingerprints of those 5 are absolutely disease-specific. So the combination of the relevant amino acids is not found on any pathogen or human proteins of the database. False positive results are a major problem in Lyme disease. By using specific antibody fingerprints as diagnostic markers, these could be excluded. And the second big question in Lyme disease is why some people or patients develop a chronic manifestation of the disease. It has been suspected that autoimmune reactions play a role in this, as the 3 specific fingerprints cannot be found on any human protein. Autoimmune reactions, at least of these antibodies, can largely be excluded. This figure shows original scans of 2 Lyme disease patients and 2 negative controls as example. In total, 7 negative controls were incubated on arrays, and minimal to no interaction was observed. So to sum it all up, for the vaccination against tetanus and for infections with Lyme disease, we were able to demonstrate for the first time that different people develop almost identical antibody specificities despite their random development and adaptation processes if the same epitope is detected. We also showed with these studies that peptide arrays can be used to accurately characterize the specificity of antibodies. It is possible to identify disease-specific antibody fingerprints with epitope mapping and substitution analysis. The knowledge about specific antibody fingerprints could be used in many different ways. Diagnostic tests based on fingerprints allow the exclusion of false positive results and the investigation of autoimmune reactions. It also allows the investigation of cross-reactive antigens. It might also lead to the identification of antitoxin antibodies for acute infections and to peptide-based vaccines. And there's another door that is opened by our studies. So for the diagnosis of a disease, you normally have to have an idea about the causing antigen. And this assumption is either disapproved or accepted with diagnostic tests. However, there are diseases for which humanity has no clue of potential antigens yet. In the future, a large group of people with the same symptoms, so presumably the same disease, could be investigated for correlating antibody fingerprints. As antibody fingerprints have shown to be disease-specific, they should correlate with an antigen of a pathogen if the root of the disease is an infection. With bioinformatic analysis, it is possible to identify these pathogens and potentially clarify enigmatic diseases. So with this, I'm already almost at the end of my talk. I want to thank all cooperation partners. Without them, the studies would not have been carried out. Namely, these are Felix Löffler, Andrea Palermo, and Frank Breitling of the Peptide Arrays group of KIT. It would also have been complicated without peptide arrays from Pepperprint. So especially I want to thank Thomas Felgenhauer in this matter. The sera that we investigated in the Lyme disease study were provided by Richard Kneusel of the Diarrect AG in Freiburg. And the sera of the tetanus study were taken by Volker List from the medical services from KIT. And for overall advice, I want to thank Ralf Bischoff from the German Cancer Research Center. Our studies were funded by several grants of the EU, BMBF, and the Helmholtz Association. With this, I thank you all for your interest in these studies, and I'm happy to answer any questions.
Thank you very much, Laura, for the presentation. Very interesting. So the floor is open for questions. And again, please use the questions box to type your query. So there are already some questions coming in, and Laura, I will read one of them to you.
Mm-hmm.
So one of the attendees asked, why are there differences between serum samples? And did you validate the peptide with the serum?
Why are there differences between different sera? Is this the question?
Yeah, that's what I understand, yeah.
Mm-hmm. Because they, of course, show individual antibody responses. And these are, if the same epitope is detected, these are very similar. But of course, in some cases, there's different epitopes that is recognized by antibodies. So this is that, or what maybe the question was about the intensities, so where there's intensity differences. And
yeah, go ahead.
Yeah, some people just develop stronger antibody responses than others.
Mm-hmm. Yeah, I don't know, maybe the author can outline this a little bit better. So I'll keep monitoring here the questions box. Also here, someone asked, will the webinar be archived for future viewing? Yes, it will. We will post it on our website within a couple of days, so you can go back to our website and check it out and review it. Give us a couple of days and it will be there. Here's another question. Please comment / clarity about nonlinear conformational epitopes. So there's someone interested in conformational epitopes. Not sure whether this has been looked at in your study. Laura, any comments about this?
No, in these two studies that I just presented, we didn't look at conformational epitopes, so only to linear epitopes. As I said, with the tetanus study, all vaccination titers were confirmed, so all sera that we examined had a protection against tetanus, but they didn't. Not all of them showed a strong antibody response with linear epitopes. So it might be possible that in this, or it's very likely that in this case, conformational epitopes play a role. It would also be worth to repeat the studies with the cyclic peptide arrays of pepperprint. It might be something that we could do in the future.
Okay. Also letting the audience know that we do provide conformational screening here at Pepperprint. Another question here, this is more now going towards the peptide arrays. Do you also provide microarrays covering the proteome of other pathogens? And perhaps we can address this to Christina Lehrer, one of our panelists from Pepperprint.
So hello, I'm from the Pepperprint headquarters in Heidelberg in Germany, and I will probably answer all the microarray related questions today. So regarding these questions, yes, of course, we can print any custom peptide set that you are interested in. So if you are interested in a specific pathogen, we would just produce a custom microarray, and we can print on request up to 35,000 peptides in duplicate on one single chip. So that already fits the proteome, at least of some viruses. But we also have a couple of pre-designed standard microarrays that, for example, cover whole viral proteomes of viruses such as HIV or Zika virus. But there's a huge list of our standard microarrays also on our website.
Great. Thank you, Christina. So another question is that one we get regularly. How long does it take to print one microarray chip? Can you also briefly comment on that?
Yes, sure. So the printing itself of the microarray takes about 3 to 4 weeks. If the customers are interested in the cyclization of the microarray, this takes another few days. If the customer is also interested in the whole assay service, we provide a complete whole report. So from the printing, the screening, and the analysis after 6 or 7 weeks in total.
Okay, great. So more questions are coming in. Let me just see whether I get the order right here. Here's another question that may be going to you, Laura. Do you think you could use this technique to strengthen interactions between specific molecules? Again, do you think you could use this technique to strengthen interactions between specific molecules?
Yeah, I think this would be possible. I mean, if you find a interaction, you can sort of, with the substitution analysis, you can identify artificial molecules that interact even stronger as the natural interaction partner. Does this, I guess this answers the question?
Yeah, we can keep it at that. And I'm going to read 2 more questions. Again, questions are coming in. And again, if your questions won't be addressed in this webinar, we will answer you offline. One of the attendees is asking, you mentioned you exclude false positive hits. Do you also exclude false negative ones?
Mm-hmm. Yeah. So in this case, the data that I just show, we can only exclude false positive test results, or at least we can investigate them. But in these studies, not all of the patients, or not antibodies of all patients, recognize the epitopes. So in case of Lyme disease, there's a lot more antigens that we could investigate to cover all patients, so to exclude false negative test results. For the tetanus study, this wouldn't be possible because there is no more antigens, there's only this toxin. However, maybe we could cover all patients or people with conformational epitopes or at least cyclic peptides.
Great. So here's another question by an attendee. It's also very fitting. It goes towards the capacity of the peptide arrays that we have. So I'm going to read this. He asks, the proteome of a virus like CMV, encompassing some 170 proteins and some 64,000 amino acids, the proteome of a bacteria or parasite is many times bigger. What is the capacity in terms of total number of different peptides addressed? So this is regarding the capacity that we have of our peptide chips. Christina, this could be one for you. Can you briefly comment on this?
Yes, sure. So as I already mentioned, we provide chips that fit up to 35,000 peptides in duplicates, which are then 70,000 peptide spots. So this fit, or a viral proteome might fit on one of these slides. But for bacterial proteomes, of course, they're a lot bigger than that. So we can either produce a number of these huge slides and perform the screening of all of these slides in parallel. Or there are also some bioinformatical options to reduce the starting content. So maybe there's already some database knowledge of more immunogenic epitopes of these pathogens, and we can also perform a database search to reduce the content.
Great. So we're going to conclude the questions here at this point. And again, if you have sent us a question that has not been read and answered, we will do this offline. So thank you all for attending this webinar presentation. We also want to thank our speaker, Laura, again. Again, we will make the presentation slides and the webinar file available on our website. There will also be a follow-up email that you'll receive. Again, if you have any questions down the line, please contact the Pepperprint team directly. Feel free to check out our website for updates, future events, webinar announcements. I will end this webinar now. Thanks again all for dialing in, and stay interested. Thank you very much.